Hello, everyone, and thank you again for the opportunity to present our recent work. My name is Julio Camarero. I'm a professor in pharmaceutical science here at the Department of Pharmacology at USC. Let me tell you a little bit of my background. I have a PhD in bioorganic chemistry from University of Barcelona, Spain. I did a postdoctoral training in protein engineering at the University of Rockefeller. 4 years, and also Lawrence Livermore National Laboratory for another 4 years. I was also a senior scientist at Lawrence Livermore National Lab, leading projects in molecular countermeasures for 4 years. And then I joined USC in 2008. So our lab is focused on developing novel molecular platforms for targeting protein-protein interactions. We are actually working on what we call ultra-stable microproteins to target these PPIs with pharmacological relevance. We use this scaffold, we call it cyclotase. This is a relatively new molecular scaffold, highly stable, that can be used for molecular recognition, has 5 hypervariable loops, and it has the potential to present oral viability. So using molecular evolution on this scaffold, we are targeting PPIs involving cancer, E3 ligases, and PD-1, PD-1, mostly autoimmune disease. We are focusing targeting BAF pathway in lupus, and also antiviral targeting the proteases required for the virus to enter in the cell. So one of the targets that we are using is targeting the E3 ligase, HDM2, HDMX. So it's well known there are HDM2p53 inhibitors. This is a validated target. Like Nulin-3, AMG232, AMI77301, but these only work in wild-type p53 cancer cells. The problem is that we have TP53 mutated in roughly 50% of the human cancers. So that renders these compounds very inactive in this particular type of cancers. So we are using a different approach. We are not targeting the binding of the ligase to the p53, but the E3 ligase activity. Which we know in this case, this stabilized not just p53, but also other several tumor suppressors proteins like RANX3, ATF3, and FOXC3A, which leads to tumor cell death in a p53 independent or dependent if the p53 is in the cell. So using in-house technology, we have developed a new cyclotide that is a very potent HDM2 and XDMX E3 ligase inhibitor. So it has an activity around 20 nanomolar, IC50. We know the mechanism of action. It binds to the C-terminal of the ring domains and inhibits the ligase activity in a dose-dependent pathway. It also is able to stabilize p53 in cells with p53 wild type. So this compound is very active in several cell lines with wild type or mutated or null p53. This is an example with colorectal cancer cell lines. And more interestingly, also has been shown to be active in efficacy in 2 animal models of colorectal cancer. So using human ACT116 in nude mice, and also in a syngeneic model of colorectal cancer CT26 cell line in biopsy. So interestingly, this compound also it doesn't have, or it doesn't show any significant immunogenicity, and has the potential, as I said before, cyclotides have the potential to be dosed orally. In this case, we have tested the viability of this compound, and it's around 30%. So we are actually funded by NIH, DoD, and several foundations. Our current collaborators are Dr. Lenz, CRC, colorectal cancer, and Dr. Ringa for lung carcinoma. So we are actually looking for funding, further funding collaborations to expand studies to other types of cancer with mutated or null p53 phenotypes, to include PDX animal models. Also explore combination therapy with other anti-cancer drugs using RNA-seq data as a rationale for deciding which compounds to use. We also like to, I just show you the immunogenicity in mice. Is not that there is no immunogenicity in mice, but we like to expand that to other animals like rats, rabbits, dogs. We probably would like to scale up the production of this compound under GLP and GMP, because our goal, our final goal is actually perform clinical trials in humans. With this, I will thank your attention.